Hi, I'm Naya, this is Ben, and this is Eric, and we're going to be talking for the third time about the biotoxicity of silver nanoparticles.
So what is biotoxicity? Biotoxicity is basically what the name implies. It is it's like how toxic a material is that can affect life, or like in this case, cells. So why are we using silver? Silver has natural antimicrobial properties, meaning that it has the ability to kill microorganisms such as bacteria and viruses and fungi. And the utilization of these health benefits can be seen throughout history, from the ancient Romans, who introduced silver into their water supply to keep the water supply fresh, to Chinese emperors who ate with silver chopsticks, and then to silver leaves that were used in World War I to treat wounds, and then up to today in modern medicine. But how does biotoxicity relate to nanoscience? So biotoxicity, the effect of silver varies greatly depending on the size of the silver that you use. To test this, we used 3 different sizes of silver. We first used a large powder, a relatively large powder. We then used a nanoparticle, which was on the nanoscale, and then finally we used an ion, which is much smaller than the powder. The large powder. Okay, the nanoparticle has a much larger surface area than the powder because it is so small. Larger surface area to volume ratio because it is so small, and the importance of that will be explained later. And then the ion, although even smaller than nanoparticle and therefore having an even larger surface area to volume ratio, expresses its own challenges in how small it is.
Okay, so just an overview of how silver is toxic to biological cells. So depending on the size of the silver, it can penetrate through endocytosis or membrane protein channels. And so once inside the cell, the silver ions take an electron from oxygen, which catalyzes formation of a reactive oxygen species. And so this reacts with the lipids in the phospholipid bilayer in the membrane of the cell and ruptures it, so it kills the cell.
So now for the fun stuff, we're going to be looking. Oh. So over here, you could see we're at a TSA check, and over here we got some bad guys who are going through security, which is very bad, and they're like the silver like nanoparticles, and you could see they pass through security. So they're supposed to, like, they're the silver nanoparticles, and once they get through, they kind of like they take off their mask, which is like the cell thinking they're glucose, but not because they're the same size, because they're pretty similar in size. And then so they go onto the plane, and then they like blow it up, and which is bad. But over here, and that was like very efficient and easy because they had like no hassle through like security. But over here we got the silver ions, which is the bad guys. They're like going through security, and then kind of the ion channel. Instead of in this picture, the equivalent would be the special TSA room, where they, suspect something's going on, and actually something is going on, but they still don't stop them. It just takes them a little longer, a little more time, maybe a couple hours or so in the real world. And then, so like they go onto the plane, and then they still blow up the plane. So that would just be like the silver ions in the ion channel. And then over here, we got someone who can't get on the plane because he's the silver powder, and he's too big to actually get into the cell, the yeast cell, and cause any damage. So then the plane is safe and fine, and the plane is not in harm's way.
Okay. So one thing the cartoon didn't address was how, in our experiment, we actually monitored the living cells to tell the effectiveness of the different sizes of silver. So we used yeast as our living cell, and so we measured its carbon dioxide emissions, since it is an aerobic microorganism and it goes through cellular respiration. And so the presence of carbon dioxide indicated like the live cell and the failure of the silver powder. So when we added silver powder to the yeast, the carbon dioxide there was carbon dioxide being produced because the silver powder was too large to penetrate the cell wall. And it did slow down because the silver powder still worked. It just took the longest and was the most inefficient because it had to go through a lot to break down into individual ions. And so when we added the nanoparticles, the carbon dioxide production halted since the cell mistook its size for food and absorbed it, so it effectively poisoned itself. And so when we added silver ions, these were the tiniest and were able to pass through the ion-gated channels, but this took a longer time. And so the carbon dioxide continued for a while, then stopped once the silver ions eventually penetrated the cell. Yeah, thanks. Questions?
Oh, yeah.
Why are we not poisoning you all with stealth bombs?
Yeah.
So we,
Oh.
So I actually looked up, there was an experiment in 2010 by the Chinese Academy of Sciences, and so they exposed human cells to the silver nanoparticles, and only 4 to 9% of the exposed cells were ruptured. And so when we were learning about this, we learned that cells can cope with the silver nanoparticles, and they use vitamins A, C, and E, and the enzymes superoxide dismutase and pyridoxase. And we also thought that we could factor in how humans have an immune system and like specialized cells to target like intruders into the body, while like yeast cells do not.
I think also part of it is that our cells are not quite as stupid and don't eat everything that comes their way.
Yeah.
Where some microbes are just,
Yeah.
it's only one microbe, they'll live or die, they'll just fry.
I think like one part of it, yeah, just like what you said with the yeast cells, is everything was very optimal for them for a reaction. So like the water was warm, there was sugar and plenty of space for them to actually expand. But in the body, it's not necessarily trying to constantly do that. And maybe one more thing is when we talk about nanoparticles and their surface area being way larger, we don't mean one small nanoparticle. We mean like if you fit a bunch of tennis balls in a basketball, that has a larger surface area because it's a lot of little particles. We don't mean one small particle has the same amount or more surface area than a larger object.
Okay, your last chance, parents, one parental question to end. Last question of the whole time.
Any questions? Maybe over there. Yeah. Yeah, we had a lot of fun.
Yeah, we had
Yeah,
so much fun.
lots of fun.
What was your favorite experiment?
I think this experiment. I think I would have to say that, but I would maybe not experiment. Maybe I think the coolest thing we've done is probably look at the electron microscopes. I think that's pretty awesome, and they were really cool. You could look at really small things. Anything else?
Then let's thank our
Sorry.
panelists.